good afternoon, everyone. So, hopefully, my I'm audible and screen is visible. So, we'll be starting with today's week six live session. So, in this week six, so till that time, we have gone through different uh, like you have seen that we have gone through different protein microarrays. Now, then we have shifted to the reverse phase protein array. So as in the lecture as shown, the different reverse phase protein arrays. So we will be yes, sorry. So we'll be using different reverse phase protein arrays and then the level 3 based techniques. One is the surface plasmon resonance. So before going on, so let's just look at that how the so you know that the as in the lecture it has been told that it will require a specific you need since it is an antibody based approach, so you need a very high throughput robust antibody, and so that what your data comes so that you are pretty much sure and your validation is quite you are very much sure of the validation that whatever you are reporting is correct, and uh, it is not beyond that. So we will look at, so that's why for checking of the robustness and the reproducibility, so in this study what they had done is then into three different platforms here refers to the three different labs. They had done the same experiment to check out that the same if the because if you have gone through the lecture and have understood the principle of the reverse phase protein arrays, so here you need a setup for the uh, so we need a particular setup for printing out the protein lysates. So from the cell lines, if you prepare the lysates or the tissue lysates, so since so the basic principle states here that you need to print those lysates. So the lysates contains the protein, and then you will be validating with the antibody. So the preparation and the printing of the chip varies from in-house platforms. So to find out how reproducible it is, so you need to have a, so they have done into the three platforms. So you can see that to assess, so I just highlighted here, so to assess your reproducibility of this RPP reference based protein array technology across multiple laboratories. So they developed an international multi-platform approach that integrated RPPA data derived from three different research sites across Europe. So the three different research sites, so integrated RC reverse phase protein array data, whatever they received, they tried to so they tried to integrate and tried to find out that okay, so that the data is quite reproducible and quite robust, so that one can rely on this technology for further uh, taking. So here you can see that they had selected 6 breast cancer cell lines. So to but you said that you can uh, so that you can take into account all the three different breast cancer molecular subtypes and different drug sensitivities. So the cells were cultured in the absence or presence of two kinase inhibitors for 20 minutes or 24 hours and lysed in biological triplicates. So there were lysed in biological triplicates. So I think you all are aware about the replicates. So one is your technical replicate, which is focused from the same samples. Otherwise, is your biological replicates, which from the different cell lines or different. Okay, so and so they generated around 108 snaptrogen lysates, which were shipped to the three different research sites and the samples were analyzed at each site using the respective in-house RPs. So the thing, as I just now mentioned, that you need a uh, reverse phase protein array in a in the it require an in-house protein platform. So it becomes quite important to measure that how much it is reproducible or robust. It should not be a lab specific or an in-house setup specific or a particular laboratory specific identification or any claim that comes only from a particular laboratory. So it needs to be accepted, and the same result should be come across uh, labs. So that's why they have tried to come across. So what all those lysates. So samples were analyzed at this site using the respective in-house RPPF platforms, and the setup of which differed at many stages of the RPP analysis workflow. So there the setups also differed. So you can check that whatever is coming would be very much robust, everywhere, including the slide type, the number of technical replicates and dilutions. So the technical replicates are those which are generated from the same samples, dilutions per samples, readout dye, scanner, image analysis software, and normalization procedure, enabling the capture of variation between RPPA platforms operated in uh, different laboratories. So as I mentioned, the also need to capture the variation between the RPPA platforms, so which is operated in different laboratories. Then microarray samples were probed with panels of validated antibodies in routine use on the three RPPA platforms. So microarray samples were probed with panels of validated antibodies. So that validated antibodies, as you are aware, that it is quite important here, so that you need get an overall idea of the protein expression in your lysates. So to enable dataset component were assigned unique antibody identifiers. Only data derived from antibodies targeting the same proteins or phosphorylated residues, including different antibodies from different acquired on all variants for further analysis. So here, so the different all antibodies were assigned their unique antibody identifier. So you can just you all can just imagine that how much uh, you need to have an so all antibodies that are having unique identifiers, and those data derived from antibodies targeting the same proteins or that were already compared. So the experimental design was very much robust for an assessment of the inter-platform concordance. So here you can see that the cell types, so they are The platform robustness can be checked from starting from that the six different cell lines types. Then their samples were prepared. So here there is no difference. The difference, so here the variation has been cut off. So it does not happen that the variation that happened occurred in the during the stage of the cell lysis preparation or during the RTPA. So that thing had been cut off by preparing the samples